Hi, good morning, and welcome to the ZP Developer Zone webinar. So we like to do this webinar every Sunday at about 8 a.m. London time, and I'm going to go forward fairly quickly. I'm looking at my internet. If it's really looking not so good, I might have to record this, but that said, let me go forward. So we do a weekly webinar. Today's the 7th of January 2023, and we're covering really questions that have come in during the week. It is relevant that a lot of questions that have come in during the week this week are really about taking assays into the field. So somebody wants to measure, for example, Troponin I, which is a very useful marker for human health, including cardiovascular health. And this is something that ZP's worked on for quite a few years. And somebody else is also quite interested in measuring water quality. And again, this is something that at ZP that we've worked on for quite a long time. So we do have hardware that really allows people to take this kind of technology to the market much quicker, or take it to the application much quicker than they would have otherwise thought. And it's called the SENS platform, and I will discuss that a little bit today. That said, let me go forward and just say we do free webinars. So for example, there's a live demonstration of SENS on the 24th of January 2024. I will put a link underneath this video when we've gone live, and this link will guide you to basically a sign up page. So if you're interested in signing up and attending the free webinar, whether you're interested in measuring the water quality or whether you're interested in measuring Troponin I, these kind of things will be covered, and you'll see the hardware in action in those webinars. Also, just to say as well that if you're a large, we do workshops on site at ZP's headquarters, and we also take workshops to other people's facilities as well. So if you're kind of interested in attending or having a ZP workshop at your facility, please let us know. These workshops are not, sort of purely lecturing based. They're very interactive, and we do things like everyone gets to build a biosensor, everyone gets to test a biosensor. We go through all the different types of modalities, and amperometry, potentiometry, voltammetry, impedance spectroscopy. These all get covered, and we also talk about kind of commercialization, and also if it's a medical application, we also talk about ISO 13485, regulatory assurance, quality assurance, and regulatory clearance. These are also essentially important parameters to cover. So we do this in our workshops. That said, let me go forward a little bit more and just say we also have a conference being held in the UK on the commercialization of electrochemical assays. This is, I think, particularly relevant to startups and to academic colleagues, so I'll just put a quick link to that as well. So question number one really is about screen printed electrodes. Somebody wants our recommendation on screen printed carbon electrodes. They're trying to detect a lithium ion. I've had a little bit of an email back and forth with them to discover where they're at the moment, and they're looking for an alternative supplier on a screen printed carbon electrode that they have, and I will discuss that today. Somebody else wants to measure water quality. They put ammonium, nitrite, nitrate, phosphate, chlorophyll, E. coli, excuse me, interstitial enterococcus, dissolved oxygen, also salinity, temperature. So we have an ammonium sensor, we have a nitrate sensor, we have a phosphate sensor. Chlorophyll A, by the way, is super easy. It's a redox molecule. E. coli, if you ever Google Zimmer, Peacock and E. coli, you will see that we've definitely done E. coli sensors. We've done a lot of bacteria and pathogen sensors, but we really only do those as part of projects. They're really immunosensors, whether you now use an antibody to detect them or whether you choose to use aptamers, etc., that's to be determined, but we only do those as parts of projects. Doing anything that's kind of more pathogen or bacterial based is more complicated. But we do have a salinity sensor, we do have a dissolved oxygen sensor. I mean, sea temperature is just, I don't want to over trivialize it, but you can stick a thermometer in there to get sea temperature. Though I think, so that's not really something we can add so much value to, let's say. But we do have the sensors, and I will discuss it. Question number 3 is about Troponin I sensing, and I will discuss that. And question number 4 is somebody wants to do multi-analyte sensing. They say 8. I'll describe to you that we've actually got quite a lot of experience in doing 6. And I'll basically sort of put that as the last section of the slides today. So screen printed electrodes, this is the easiest one, and I will just describe it quickly. That at Zimmer & Peacock, we do have probably, well, easily over 40 screen printed electrodes. But over the years, I've kind of realized that, you know, when you try to do so many screen printed electrodes, actually you're doing a disjustice, because you've, in order for something to be really effective in manufacturing, that is, you need to focus. If you try to manufacture and you have broad range of manufacturing, it actually makes it a bit tougher. So we focused our screen printed electrodes down to a few that I really, well, we do all of them, but these are the ones we do in high volume. We've got this 501 carbon. This 501 carbon is a high quality, low cost sensor. When I say low cost, I'm super suspicion that we do about 300 of these electrodes for about 268 euros, so less than €1 each. There is a platinum version. Platinum is much more expensive. I know this person's asking about carbon, but just a general comment. If you can avoid platinum in your electrochemical sensors, your electrochemical assay development, avoid it. It's a really expensive material. It's not going to get cheap. It's a dead end material, I think, when it comes to answering the question, how do I make a low cost sensor, or how low cost biosensor? Hypervalue carbon electrodes. Of this range of electrodes I'm showing today, this is the one that we've had the longest. This is the one we make in the highest volume. I do just feel like for my academic colleagues, they like, so at ZP we have robots to do our pipetting, but academics, they often use hand pipetting, so they prefer the circular electrode because you can sort of put a drop on there and it spreads out. They're quite happy. But in fact, I also quite like this hypervalue carbon electrode. There's a platinum equivalent. If you can avoid platinum, avoid it. Gold is exactly the same these days. Once upon a time, platinum used to have this reputation for being more expensive than gold. That's a long time ago now. Gold is super expensive as well. I know I've said it already. So if people who are trying to think about cardiovascular, for example, biomarkers, you will find papers that try to do this on gold, and you'll find a few papers that try to do it on carbon. Please just do it on carbon. You will have such a different cost structure when it comes to carbon versus gold. We can do carbon screen printed electrodes for less than €1 each. Gold ones, we're doing them about €6 or €7. It comes down to a fundamental that the raw cost of materials for gold and platinum is about 27 or 28,000 times more expensive than carbon. It just starts with that. It's the start of the problem, and it just gets worse from there. So I recommend to this person asking about carbon, I can't recommend it more. I'm also going to give you evidence as to why I recommend it more as well. There's 501 carbon here, and I'm also going to give you a recommendation on this hypervalue carbon here. But because of who you are and where your situation is, then possibly the 501 is the one to go forward with. I'm now going to, so the 501 carbon, if you see a link jump up, the link will be underneath the video, and hopefully you can navigate and find it. 501 carbon, 501 platinum, which I'm not recommending. The hypervalue carbon is a good electrode. It may be just harder for you to pipette upon. Hypervalue platinum, hypervalue gold. You will need cables, so depending on what potentiostat you have, you will need, obviously, cables to get between your potentiostat and the ZP connector. And I bring up the ZP connector here because I've just come across it that people don't have the right connector with our screen printed electrodes. And so you get distorted data, and it's nothing about the quality of our screen printed electrode. It's all about the electrical connection to the screen printed electrodes. We do talk about, the reason I bring this up is because I've suggested that people come to our Sense It All webinars. Whenever you see Zimmer & Peacock, whether we're working on developing medical diagnostics for things like cardiovascular, we always try to avoid calibration. And the reason we can avoid calibration is often because of actually the underlying repeatability of our screen printed electrodes. This just makes us really different from the competitors. So if you're interested in a screen printed carbon electrode, many of our competitors, they make that screen printed carbon electrode for you. They're not consuming it themselves. So at ZP, we're very unusual because actually the screen printed carbon electrodes that we use, we then go on to use them in our own products. So we really have to care about the quality, and it's a very different mindset. So we really do care about the quality. So what we're doing is we're doing voltammetry on the batch. We're sacrificing a proportion of the batch that we're making to make sure that they're actually repeatable. I think the biggest problem is with carbon screen printed electrodes is not necessarily the individual quality of the electrodes, it's actually the repeatability of the electrodes. So if you're trying to make a sensor, if you don't have a repeatable substrate on which you're making your sensor, you will not have a repeatable, essentially, sensor. So then you start getting into people who are normalizing the signals in academic papers. People are probably just rejecting data a lot in academic papers so they can end up with a nice linear graph. So there's a lot of sort of data manipulation because fundamentally what's happening is they don't have repeatable electrodes. Well, at ZP, if you want to make a sensor repeatable, if you want to make a sensor useful, you kind of have to have a relative standard deviation of less than 10%. So at ZP, we check the entire batch. We look at the relative standard deviations 6, 5, 6, and 6, and make sure that we think that these electrodes are fundamentally reproducible enough. And so then when we're doing our work with these electrodes, here I've used voltammetry. These are all the concentrations are done in triplicate, but actually we get a really nice sort of narrow distribution in signal because of the fundamental reproducibility of the electrodes underneath it. So my biggest recommendation is that 501 carbon. Why? Because at ZP we really actually do use our own screen printed electrodes in our own products, in the Sense It All platform, but also in something called the Food Sense. We use our own products, and so it's the first people to know whether there's a problem with our screen printed electrodes is ZP. So watch out. So that's why I recommend the 501. On potentiostat front, one of the biggest things I have with modern potentiostats is that they look like scientific instruments. The whole thing about electrochemistry is it's a really practical science that can really impact society. So if you want to measure water quality, screen printed electrodes and biosensors is a really good solution for it. If you want to measure cardiovascular, or let's use diabetes. So when you look at diabetes, it's treated with meters and strips. Those, at least a proportion of those strips are all screen printed. Most of the home diagnostics for diabetes today and the hospital-based systems are all electrochemical. So electrochemistry really demonstrates its ability to actually leave the lab and get into people's hands. Problem is, potentiostats are large, bulky, they're the wrong form factor, they don't inspire people to actually realize that what they're doing is very translatable. So we actually have a potentiostat called Kate. It's beautiful. You'll see some more videos in a bit, but actually Kate is a fully functional potentiostat. But it's also, with a software change, our Sense It All platform. So what Kate allows us to do is to have people doing research on hardware that actually can then become a real product. And it really eliminates the gap between what you do in the lab and what you can actually practically give to people in the field. So take a look at the Kate potentiostat. If a link jumps up, that link will be underneath the video as well. And there's also a link here to Sense It All as well. Just a side comment. People who are trying to commercialize technologies, whether it's for cardiovascular or whether it's for water quality, the Sense It All, when we're doing it in volumes of 10,000 units, so it's a large volume, so I'm not expecting universities to do that. But when we can get to volumes of 10,000, we're able to sell it at $60 each. So it completely makes sense with a business case as well. So get yourself onto the Kate potentiostat as quickly as possible. Just something that, one of the workhorses that we do use in our own lab, by the way, is something called EMMA. EMMA is a parallel multi-channel potentiostat. This is also relevant to the people in something like question 4 who want to do 8 channels. This is actually a 6-channel potentiostat. Got a nice little touch screen on it. And this is kind of what we use. The reason I bring this up is because when you're trying to do any kind of development work, you need to be running sensors in parallel. Otherwise, you just don't have enough statistical data to make good either academic research or business decisions. You need enough data. To get enough data, you need to be able to do it all in parallel. So just take a look at the EMMA potentiostat as well. Question number 2 is about water quality. I'll just have a sip of coffee. Okay, so in those water quality, You're interested in ammonium, nitrite, nitrate, phosphate, chlorophyll, E. coli, this enterococcus, and salinity and dissolved oxygen and water temperature. One thing I quick say is, we do have a nitrate sensor. We don't have a nitrite sensor. I'm wondering how quickly nitrite gets oxidized to nitrate in seawater anyway. But that said, let me go forward and tell you what we do have. So we do have a whole bunch of biosensors there. I think they're in the right. By the way, our biosensors can be either used discretely or continuously. And we have ammonium, we have nitrate, we have phosphate, we have conductivity, and we have oxygen. You didn't ask about pH, by the way, which is also kind of could be also quite interesting to you as well. There's probably a few more in here as well that you might be quite interested in. So we do have a platform for it. SenseIt is a platform when really it's we describe it as sensor to cloud. What this means is if you measure something in the field, the data will end up in the cloud. So I'm supporting people around the world at the moment on making measurements. And what makes that support so good is because actually I can see their raw data arriving in the cloud. They've decided, it's their choice, to share their data with me. They've invited me to see it. And because it's all going to the cloud, we have a cloud system called Julie, by the way. If you're unclear about that, come to our SenseIt webinar on the 24th of January. So we have our hardware, we have our we have this SenseIt electrochemical platform. We also do wearables as well. So we're quite into kind of continuous measurements on people. And we are doing a lot of environmental type applications these days as well. Not so important, but we've got things like nitrate, phosphate, potassium, pH, ammonium, urea, calcium. These are the kind of things that sort of tend to turn up a little bit when we're talking about environmental or agricultural applications. So the whole philosophy of SenseIt, and this, you can, whether you're talking about agriculture, environmental or medical, is you've got somebody with a need, so it could be an environmental monitoring system, and you've got people who want to serve that need, and that will be yourselves, academics, entrepreneurs, what's called entrepreneurs, companies. And so SenseIt is a platform that kind of quickly gets you from your idea to the person who needs essentially the help or needs the measurement. This kind of process is kind of, the philosophy is minimally viable product. Too many people spend too much time doing too much research and never comes out of the lab. Well, at ZP we say, here's the sensor, here's a piece of hardware. You'll see the app for this in a bit as well. And actually you can give that hardware to some people, real people who have real problems, let's say. They can start testing. And then the data automatically goes to the cloud. So we have an app on the available on the Android store and also on the App Store called SenseIt. You'll see that in a bit. And that app will send all the data to the cloud called Julie, and then you'll be able to see the raw signal in the cloud. And like I say, I'm doing this at the moment for somebody in California where I'm literally looking at his data in real time and able to kind of help him and guide him into actually getting accurate results. The cloud also has an API on it. This is an application program interface. This is a way of pulling data across to your own clouds. We're able to improve the algorithms and the sampling and of techniques to actually make this more accurate. So we have a kind of a lean startup philosophy. If you're not aware of lean startup is basically a way of sort of saying, get to minimally viable product, get something tangible quite quickly and get it in the hands of people quite quickly so that you can find out what you really, is what you're developing even actually useful. So many programs go for years and in fact what's developed is not even that useful. So that's our philosophy at ZP. That would be our philosophy for doing those analyte testing in fresh water. And maybe that webinar on the 28th of Jan would be useful. This might be quite useful as well. This is the actual hardware itself. So what's quite good about this piece of hardware is you'll see me take it apart in a bit. So if it gets a little bit dirty, a little bit contaminated, if you're testing blood, you can wipe it down. If you're testing seawater and you get a bit of sort of seawater dried upon it, you can wipe it down, and you'll see that in a bit. Here, the scientist is actually connecting to the hardware using the app. She's selected the app, so the smart device found the app through Bluetooth. It's got what's called the MAC address of the device. She's putting in the details. Now she's going to actually run the assay itself. So she puts the sample on there, and then she runs the test, and then the raw data actually goes up to the cloud system called Julie, and we get the raw data up in the cloud. The last video, I think, is super useful for the guys who are thinking about doing water quality testing, because for every water quality test, there's a QR code exists, or and so you scan those QR codes through the app, and the app will then reposition itself or reskin itself to deal with that particular analyte. And it all does it off this particular piece of hardware. And you can see her hands here, and you can also see my hands here as well, how small this piece of hardware actually is. So we do a lot in the agriculture and environmental applications: redox, pH, chloride, potassium, calcium, sodium, phosphate, nitrate, and ammonium. These are the sort of things that come up. But every time you take one of our sensors and put it into a new application, there is some work to be done. So don't think this is for free. You really have to do quite a lot of science to make this actually work. So for every application, there is a development effort. So don't think you're giving a turnkey solution here. You have to be involved in the development of these things. But it's nice, I suppose you can think about the technology readiness level. It's nice that actually you can get that app off the Play Store here, or for the Android store here. And if you do let's say register, here's a chili application, here's a ginger application, here's a nitrate application, and here's a total antioxidative status application. So when you've got the app installed, either off the Apple Store or the Play Store, the first thing it'll do is ask you to scan a QR code. I just want to say good morning to Ali. You can scan this for chili, you can scan this for ginger, you can scan this for nitrate, and you can scan this for TAS, and that'll give you an example of how to go from these different analytes. But I just want to say good morning to Ali. Now the other thing I want to, this is just the last thing, just to talk about the workflow. So this person wants to measure water quality. So we have sensors, we have hardware, we have an app, we have a Julie database. So you put the sensor into the hardware, you put the sample onto the sensor, the app will run the meter will give the data back to the app, and the app will send the data to Julie. And in fact, you will get a result on the phone there and then. And if you watch our demos, our live streaming demos on the 24th of January, you'll see that we actually get the raw data. This is very useful because whenever you're first going out to alpha testers or beta testers, people make mistakes. You have to find out what those mistakes are. So you have to go and look at the raw data to make sure everything's looking okay. And then you can actually share it with people like us. So you can actually get some, like, a real, let's say, some pretty good world-class scientists and engineers around sensors also looking at the data to make sure that everything is making sense. So that's the kind of workflow that we have built into that. And again, come to the webinar on the 24th of January. I'll put the link in, and that will help. Slight change here. So we've been talking about environmental sampling. Now I want to touch upon medical applications. So at ZP, first of all, we are ISO 13485. So we're certified for the contract development, contract manufacturing of medical devices, including medical diagnostics. We've got a long experience. So I started my career in 2000 working for Abbott, making glucose strips. So when you look at a lot of technology from ZP, what you're fundamentally looking at is something that looks a bit like at-home glucose strip testing for diabetes, because diabetics monitor themselves with glucose strips. So I obviously started this kind of 23 years ago doing this kind of work. The troponin sensor is more complicated than a glucose sensor. Glucose is intrinsically high concentration, 5 millimolar, whereas troponin I is actually quite low concentrations, talking about picograms, maybe some nanograms in concentrations per mil. Also, a glucose sensor is enzyme based, a very robust enzyme, whereas troponin is more immunoassay. It's actually more complicated assay and more sensitive and delicate materials to make it. But actually, we can actually implement the Troponin I sensor on our SensiOR platform. And the reason I would suggest doing that is because it very quickly goes from a sensor in the lab to a sensor that can actually be started to be tested with real samples. And so it really minimizes the gap between an idea really and starting to get what I would call preclinical data. You find so many companies I've seen companies do 15 years of actually development and still not be successful in medical diagnostics. So I think it's better to have a 3-month to 6-month sprint and get some hardware out to really help, or to start giving to the actual doctors. So for example, you have a doctor, Troponin I, for example, is a known cardiovascular marker. We can implement it on our SensiAll platform, and that can actually then go out to the doctors. Now what's nice about that then is all the data goes up to the cloud. You don't have to put the patient data in the cloud. Let's be clear about that. There's a big thing, especially in Europe, about putting patient data in the cloud. Putting raw signal in the cloud is not putting patient data in the cloud. It's putting raw signal in the cloud. And as long as you don't put patient name, ID, birth dates and stuff like that, things that would identify them, then you're fine. And the hardware doesn't require patient data. It just has a test number. So test 21, for example, it's very generic, and the raw data will go to the cloud. So there, you can actually analyze it then in the cloud. We do have this API, this application program interface, and it allows us to kind of look at that raw data in the cloud and to essentially improve upon it. So to get involved with this, ZP probably needs to do what's called a proof of principle study, so we can take our hardware, talk to you a bit, have enough money or enough funding to actually talk and have that decent conversation. So I think a minimum with us would be a proof of principle upon that. If I say something like proof of principle, then I'll put a link also underneath the YouTube video as well. Don't forget our SensiAll webinar on the 24th. This will be pretty useful to you. Question number 4. These people want to do a custom, they say PCB here, printed circuit board. Whether you do printed circuit board, whether you do screen printing, to be honest with you, that's not the gating item at the moment. But we could do it in PCB format if required. They want 3 electrode systems. So let me just talk about, they list out some analytes and they want CSV, comma separated variable outputs. To be honest with you, we just put the data to the cloud. You can download it. It's interesting actually that you'd ask for comma separated variable, because actually everything should be cloud enabled from the get-go. It should all be cloud enabled. So CSV is not really a problem. But some of those sensors you want: glucose, hydrogen peroxide, lactate. Maybe you mentioned pH as well. We do have those sensors. We have been working on this kind of handheld device for quite a while. I'll let this video play itself out. So handheld technology, where you have one sample, but you want to do multiple analyte testing on it. I know the guys who are thinking about water quality will be really interested in this. But to do this kind of work, it really requires big budget. So if it's an academic R&D project, stay with the SensiAll platform, because that's pretty good to go. But you can see that ZP is kind of ahead of the curve in terms of, at least here we're doing 6 analytes, not the 8 that you're asking for. But we do have the capability, of taking many of our sensors, putting them into this kind of multiplexing format. Here's one sample, and the sample then gets distributed among these micro wells, and we can do testing upon those. So you can do parallel testing like that. So we do have this kind of technology. Let's say this is one of our scientists actually testing. What she has here is a row of 6 sensors, and she's testing them to make sure that everything works okay. It's interesting actually, because I realized that, of course, I know you want 8 and we've got 6 at the moment. We can take it to 8, but it's going to require some engineering, and that means cost. It's more cost. It's cheaper to adopt 6 than it is if you want 8. Then we have to basically extenuate out to 8. This is Emma. You can see Emma here is 6 as well, so you can get a sense that actually ZP has multi-channel instruments. One of them is 1. So that's the SensiAll platform that I've talked a lot about. And then when you look at the Emma potential stat, you see it's 6. The hardware I was just showing you a minute ago, you see it was 6. So you can kind of see the commonality, that what you can see what we already have, let's say, in place. So there's a quick link to Emma as well. Cheapest way of doing this is actually just use the SensiAll platform and do 1 analyzer at a time. There's a good business reason why you want to do 1 analyzer at a time. If you want those business reasons, you probably have to engage with ZP to give you business advice, but that's your choice. But definitely come to the webinar on the 24th of January. So I think that's a summary. I want to say a special thanks to Ali, because Ali's really been a good friend to us over the years, so appreciate that. Question number 1, screen printed carbon electrodes. That carbon 501, sorry to say it, best screen printed carbon electrode in the world. The circular electrode allowed to use it quickly by pipetting onto it, for example, and we use it in high volumes ourselves, so we make sure it's pretty good. We can do most of what you're asking for. At least you've got ions and small molecules and pathogens. Ions and small molecules, we can do most of what you're asking for in the Sense It All platform. The pathogens, these are projects. We would have to do what's called a proof of principle project on those. Regarding Troponin I, we have the sensor. We've tested it in spiked human plasma, and we have the hardware. If you want a glucose strip-like technology, we have that platform. Minimum start with us would be a proof of principle. And then with these guys, it just happens that we could do 6 much more easily than 8, but we could do 8 as well. And I just gave you some confidence that we have the sensors and we do have the hardware as well. So I want to say thank you very much for attending this morning. We've had a nice attendance, and special thanks to Ali. If you have any questions for ZP, as always, please don't hesitate to reach out to us. But thank you so much. Take care. Bye-bye.